Welcome to the Chapter 17 wrap-up. So at this point we've got the DNA in the nucleus. That DNA is going to undergo transcription and become RNA, specifically mRNA. tRNA and rRNA, they tend to kind of stick around, whereas mRNA is the most ephemeral, the most temporary of those, so we normally focus on mRNA when we talk about this. So that's going to go through and be produced. Now, if it's a prokaryote, then we move directly to translation. If you're a eukaryote, remember we go through this processing step before the RNA leaves via the nuclear pores and makes its way to the cytoplasm. Now, once the mRNA is in the cytoplasm, we're now open and available to a family reunion of the different types of RNA. So we've got mRNA, which is ultimately going to have the codons, which contain the code. It's going to have tRNA with the anticodons, which are important because the tRNAs are the ones that correspond the codon, thanks to their anticodon, the complement, to the appropriate amino acid that we should be involving. And so this one helps to make sure that the code of nucleotides properly gets transferred to the code of amino acids. And then lastly, the rRNA makes up the ribosomes, which are going to kind of oversee this process. So I always think of them where if you've got a younger child or perhaps older, where they'll read and they'll use their finger to kind of go word by word by word to make sure they don't screw something up, to kind of make sure they're reading it properly. Now on the ribosome, we're going to have 3 sites, and we'll call these the ape sites. The active site for the A, that's where the tRNA binds, it's the exposed codon. The P side, which I call the polypeptide site, that's where the growing polypeptide chain is, the growing amino acid chain. And the E site is just the exit site. Now, depending on which way we're going through this process, so for instance, if we're taking this process and going to the right, it'll look like EPA. If we were doing this process instead to the left, it would look like ape. So just be aware that depending on our frame of reference here, it may look a little bit different. Now, the process we're going to be after here as we watch this is a really visual process, right? When I talk about translation, it's difficult for me to just say, oh yeah, ribosome, 2 subunits, clamps on the stuff, codons. So I want you guys to watch this. So this is a good animation I found that'll take us through the basics so you guys can hopefully get a feel for it. So right away you can see that there's this GTP, that's just going to be the guanine 5' cap. Then you've got the poly A 3' tail, so you can see AAAAA. Now there will not be a promoter here, there's going to be a start codon. And that start codon will be AUG, so it's right here. And then there's 3 possible stop codons. I believe the one we're going to use is going to be UAA, which will be right here. So this is going to be a very, very short sample. In reality, this would be hundreds of amino acids long, but for the sake of time, we're going to focus on one that's like 4 amino acids long. And at that point, you should hopefully get the idea. So to start this process, the small ribosomal subunit is going to come bind at the start here. All right, you can see AUG is kind of smack dab in the middle. We're then going to have the tRNA that corresponds to AUG, so in this case UAC, and it contains an amino acid, methionine in this case. So all proteins ultimately start with methionine originally, although in many cases when we're processing this protein, we'll remove it. So in reality, a lot of them don't. But when you're first building them, they all start with that. Much like if you're pouring cement, you put in wood frames, and then you pour the cement, and then you can pull the wood frames out. So it doesn't have to be there at the end. And then the large subunit, once the tRNA binds, bringing methionine, the large subunit clasps this. And so you'll see at this P site, we now have our tRNA that contains our first amino acid, methionine. Now next to it is an exposed codon, AAG. So UUC is going to come in and bind to it, bringing its own amino acid, in this case lysine. And once this happens, it triggers the ribosome to shift, and it triggers the amino acid of the guy that's at the P site to be dumped on the guy who's at the A site. At the same time, the guy who's at the A site, this ribosome shifting, so he is now going to be sitting at the P site. So if I jump forward here, you'll see this amino acid shifts, so it's now holding its own with the previous one on the end, tacked onto it. And then in the process, this ribosome is going to shift, so it's now at the P site, holding the growing polypeptide chain. Now this means the original tRNA is at the E site for exit, so it leaves. And once it leaves, there are enzymes that are in the cytoplasm that will reattach the appropriate amino acid to recharge it so it can continue to be used. Now we've got an exposed particular codon here, so the anti-codon will attach, bringing its amino acid. That triggers the same response where we move over the amino acid chain onto the new guy, and then he moves to the P site, and then now that kicks out a guy, the next guy comes in, and so this process will continue until we reach the stop codon. Now, as we said, we're doing this much briefer than it would be, so we're going to hit the stop codon here, and then a releasing factor is going to come in that's going to yoko this whole thing up. It's going to split everybody apart. And so this chain is going to now get sent off, and it's going to go by itself, and this would then be folded, because protein structure, to get there, you have to fold it. It can be activated. We can kind of snip it and manipulate it a bit before it's done. So don't assume we have a protein. That's a misnomer here. We have a early polypeptide chain, which is going to undergo a lot of processing in terms of folding, possibly activating, possibly removing the methionine, etc. And then, even when that's done, it may be part of a protein that has multiple polypeptides, so we'd have to pair it together with the other polypeptides that it needs before we get a finished protein. So don't assume at the end of translation we're just done and we've got a protein and everything's wonderful. That's not the case. And then the rest of these pieces go float away, waiting for them to be needed again. So these ribosomal subunits can go find another mRNA, or the same one, I suppose, and they can reattach and continue this. The tRNA can be reused as soon as it's recharged. It needs an amino acid again. And the releasing factor can float around, waiting once again for when there is a need for it to attach to a stop codon. So we started at the start codon. We went through and translated, so tRNA converted between the nucleotide codons and an amino acid. So we went through this whole thing till we got to the stop codon, and then at that point we were left with the polypeptide chain and the rest of our stuff being more or less floating around like it was originally, back to normal. Now, one of the other pieces that we've got that I want to make sure that we're comfortable with is I want you guys to realize that when this process occurs, it's going to occur in many cases, well, I'm going to cover this first because I feel like it, multiple at a time. So my analogy I've usually used is kind of like the water slide, where if you have one ribosome going through and reading an mRNA, as soon as that guy starts on his way, you can have somebody else start behind him. You can't have two ribosomes at the exact same point on an mRNA at the same time, but there's nothing saying, like you can see over here, that I can't have 5 ribosomes on the same mRNA at the same time. They're just in different spots. Just like if you have that really big water slide or something, you can ultimately send a person down every 5 seconds, even if it takes 30 seconds to go all the way through the slide. Just like on a roller coaster or something, they could have multiple different carts and multiple different sets of carts on the roller coaster at the same time. The only problem you have is if 2 people try to bilocate. That's when you'll get a big deal, but that shouldn't happen with this. And then just terminology, initiation is the part that we mentioned where the small subunit will ultimately pair up with the tRNA that is the corresponding one for the start codon, and then it'll also have, then the large subunit will come down and bind on it, and so these 3 guys are all there together. So starting tRNA guy, the small subunit, and then the last guy to the picture is the large subunit that kind of clamps on, and then the process goes along. And so this elongation or translocation idea is just as it goes along, so it's not this binding part it's showing, it's this part, until you get to the termination where the stop codon makes everything break apart. So you can see here it kind of goes back to how it was at the beginning. The only difference is we've gained this polypeptide, and as I said before, that polypeptide's not fully done until it folds. In some cases it has to be activated still. There's still additional bits to be done. Don't just assume, ta-da, everything's over. There typically is at least a little bit more that we have to deal with. I just have to go through a couple ideas here. One of them is I want you guys to realize that for what we have, we've got this idea that there are 4 base pairs, 4 possibilities, and we read it in 3s. So 4 to the 3rd power. So there are 64 codons that are possible. Now, of those 64, keep in mind that we have 1 of them that's the start codon, and we have 3 that are stop codons. So there's really 60 of them that are going to actually code for amino acids. But there's only 20 amino acids. So this is what we call a redundant code. A redundant code is going to be one where there's multiple possibilities for each particular amino acid. So in many cases, when you look at this chart, which shows you how to figure out, it's basically the genetic code. So it shows us that if we look at an mRNA that has UCU, UCC, UCA, or UCG, all of those will code for serine, a specific amino acid. You don't have to know the exact name. I'll always give you the chart, but you should be able to read the chart. And so what I did is the first letter is always this first thing. So the first letter is U, so it's going to be here. The second letter, let's say it's UCU, would be in the UC column. So it's a little bit like a Punnett square where you kind of align yourself in the columns. And then lastly, you've got it's going to have the second guy, which is a U, so UCU, serine. And so if I tell you that this is the particular sequence we're dealing with, where it's something like UCU, A, GC, we can do C, C, C for the sake of simplicity. So you guys can go through and realize, okay, this is going to be a serine, then AGC will be A, and then it's, as I try to read here, AG, so it's going to be somewhere in here, and it's AGC. So that one is also going to be serine. All right, that shows you how redundant it is. I managed to pick one twice randomly. And then we've got C, 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 so that one's proline. At least we got somebody different here. So these three amino acids that we would get in this chain would be serine, serine, proline. And so the significance of this is, first of all, the third guy you have oftentimes doesn't really matter. And we call that wobble. That I could have UC anything and I'm still going to get serine. And so this is part of that redundancy that even if we have mutations, so long as the mutation's in the third nucleotide of a codon, oftentimes it doesn't matter. It doesn't change any of the actual protein structure that we get. So wobble's just going to explain why it's in some cases a little bit more difficult than you'd think to have a mutation be a huge deal. Not that mutations aren't necessarily going to be problematic, but this means that a lot of mutations won't actually change the protein structure overall. This chart over here is just another way of doing it. You can see that this is the first guy, the second guy, and the third guy. So I like this one a little bit better. I don't normally see this chart as much, but if I gave you something like CAU, you just go CAU, histidine, I believe is what that is. And so this one's kind of easy, and you can see here we've got the three stop codons. So it's UGA, UAA, and UAG. So it's always a derivation of those U's, A's, and G's. Whereas AUG is always going to be our start codon, which also codes for methionine. That's an amino acid that starts every protein, but a lot of times it's removed before we're done. So don't assume every protein actually starts with this. So that's a redundant code. Now, the last bit we have in this chapter is going to be good old mutations again. And so as we start mutations, first we should describe mutagens. Sometimes they can also be carcinogens, but these are things that cause mutations. So this is things like radiation. This is going to be things like, UV type of radiation. This is a lot of chemicals, like if you cigarette smoke and things like that. These are things that get in there and mess with our DNA. And so they lead to mutations, which can obviously also lead to cancer because that's caused by mutation. Now, of our mutations, we're going to focus on, in this section, small mutations, not chromosomal mutations. That's chunks of things. That's a tremendous chunk at once. So we're going to focus more on if we add a few things, what we'll call point mutations. You'll see that next slide. There's more of this. But this is where we're going to like swap two guys, or we're going to add a couple nucleotides, or we're going to delete a couple nucleotides. So to start off with some of the kind of adding or deleting bit instead of substituting, we'll have base pair insertions or deletions. If I do this in threes, it's not as big of a deal in the sense that at least all the other amino acids are as they should be. If I add three things, if I add a codon, I only change one amino acid, I add it in this case. If I delete a codon, I only remove one amino acid. So maybe the protein could still do something useful. However, if I were to add something like 1 or 2 nucleotides, everybody after that insertion, or if I delete it, same thing, everybody after that deletion gets screwed up. Because you actually change the reading frame is how they call this. So if our normal thing, if you read it in codons, is the old man and the sea. Wonderful. You know, that's what we're going to get. But if I go through and add one guy, in this case we just put X, it's just whatever, you'll see now it's the, Zol, Dma, Nan, Dth, Essa, A. So everybody after this screw up is messed up, every single amino acid. So these are very, very damaging mutations, especially if they occur at the beginning of a gene. If it was right at the end, I might still have the old man and the sex. That just got awkward. But ultimately, if we get this one that's at the end, it has much less of an impact. But a frame shift in the middle or beginning of a gene is devastating. And you can see below it shows if you remove it, so in this case the O's gone, and still everybody after that gets screwed up. And so anytime you can avoid a frame shift, it's going to be typically a very, very nice thing to do, because they're one of the worst things that we can possibly get when we're talking about mutations. Now the other mutations are going to be substitutions. So in this case we normally have TTC, that's the DNA, which then if we go through and we transcribe it, you'll see it's AAG, no U's, so I guess we wouldn't know for sure it's mRNA just by looking at it. And that typically codes for lysine. All right, it's a particular amino acid, no big deal. I believe that's the one in Jurassic Park that they were supposed to have made them that they had to eat or else they couldn't live. That didn't work out so well. Just watch Jurassic Park 2. Now, the three ways this can pan out. One of the ways is we change this third nucleotide and the wobble kicks in, and so we still get the exact same amino acid. No change overall in what we get at the end. So functionally, we got the same protein, no harm, no foul. Now the other thing can happen is called, and I'm going to skip here, is missense. Now in a missense, what's going to happen is you're going to change it, and so it is going to change one amino acid. Every other amino acid is fine. This substitution only affected this TTC codon, or AAG, I suppose. We should use the mRNA. We normally do when we're doing the code. So if AAG becomes AGG, we're going to get arginine. Or we're going to get, if we make it ACG, threonine, I believe is what that is. And so when we do this, this will have some effect typically, but it's usually not devastating. Especially, and you don't have to know this conservative, non-conservative, but the idea is sometimes you can actually have this where you get another amino acid that's like the first guy. So in this case, you can see some are basic, some are polar. So if I swap a basic guy for a basic guy, even though there's a change, it's probably going to have little to no effect. If I swap a basic guy for a polar guy, I'm going to have where it's probably going to have a bit more of an effect, but it still might not be huge. You might still have an enzyme that functions, maybe just not quite as well, but it's not as devastating, or certainly not as assured to be devastating. Whereas a nonsense mutation is a lot like the frame shift, where this one's bad, because you change something that was an amino acid into a stop codon. So if this occurs really just about anywhere, but especially in the middle or beginning, we now get where most of the protein's not even there. And so if it's not there, it can't help you. And so this protein's normally going to be gibberish. It's going to be worthless. And that's why it's called a nonsense. This isn't just a little slip. This is devastating. And so those are our base pair substitutions, and that kind of wraps up this idea of mutations that we can have. If you have any questions, make sure you guys ask me on Monday, and I hope you guys enjoyed this. Take it easy.